Good morning. This is a recording of Chapter 5 Macromolecules in our BIO101 section. Okay, so as usual, the first 2-3 slides that I fit in would have a preview of what is to come in this chapter, or all the things that you have to know. So I list them out. Okay, so we start here. Whenever I start this particular chapter, I ask my students about whether they have ever hit the gym and what have they come across as information or read or been told by someone when they have decided that they have to lose weight or build muscle or carry out correct dietary practices. So the first thing that they would talk about is they usually look at the nutrition facts, and then they look at serving size, they check the number of calories, and then they are also told that you have different things that are present in this particular food item. It could have something known as a carbohydrate, it could have something known as a sugar, it could have a protein, it could have fat, of which some may be saturated, some may be trans, some have cholesterol, and then it has certain amount of salt in the form of sodium or calcium and other elements, and then it can have some amount of vitamins. So in this chapter I would be talking about the main items that are mentioned in this in detail, about what is that group called, that is the group of carbohydrates or the group of fats or lipids, okay, or the group of proteins being called, and why do they come under the bracket of being a macromolecule, and so on and so forth. So the first thing that I start with my class is to generally check with them that what do they understand when they see an entire brick wall. They know that it is comprised of smaller bricks. So the first thing that it dawns on them in this particular situation is they are very clear that it is made up of smaller individual pieces, like looking pieces, similar looking pieces. They are joined together, put together in a certain fashion to give you an entire wall, right? Okay. Or if you look at another analogy, like a string of beads you have, or a string of pearls, you have same looking alike, similar sized pearls that are joined together in a string of beads. So my point is, in order to understand what is a polymer, polymer is a set of a lot of monomers. A lot of monomers are put together, and then you get a polymer. This is like a generic word. So in this particular case, this particular wall is put together by a set of bricks. So the bricks become the monomers, individual bricks become the monomers, and the polymer is this entire wall. Or you could say the individual monomer are these individual beads, and the entire chain or string is your polymer. Got that? Okay. So we have 4 critically important large molecules that have to be considered in this particular chapter. Macromolecules. The word tells us macro means huge. So these molecules are really, really huge molecules. Okay. So my point is, you have 4 huge molecules that you're going to be considering. One is a carbohydrate, another is a lipid, third is a protein, and the fourth is nucleic acid. Now, in this case, you have to remember that the carbohydrate, the protein and the nucleic acid are polymers, okay, and lipid is just a huge macromolecule, huge molecule, okay. It is not a polymer. Okay, so we then go ahead and try to talk about or find out why something is, no, sorry, I apologize. How is something put together, and how will a monomer be joined together in a repeating fashion to give you a polymer? How is this joint done? How is this link done? Okay, so for that we have to understand two things. One is loss of water, and the other is hydrolysis or breaking of water. Or loss of water or removal of water is dehydration, okay, and breaking of water is hydrolysis, wherein the water gets added to the reaction. Okay, let's look at that. So the point is, if I have a short polymer, okay, like this particular polymer that I have here has got 3 monomers attached to it. Okay, so it has this one hydrogen present here, and the unlinked new monomer that has come into the picture has one hydroxyl group. Now I am guessing my class you understand what hydroxyl group is. All of this was covered under chapter 4 when we were talking about carbon and the different functional groups. Okay, so hydroxyl group is one of the groups, and it is chiefly represented by alcohols. Okay, so an aldehyde will be a carbonyl group. Ketone will be a carbonyl group, but it will also have hydroxyl groups. Then you will have carboxyl groups, which is an acid, or an amino group, which is an amino, and you can have a phosphate group. So you were I'm kind of brushing you over the different groups that we have already worked on. One was the hydroxyl, and then a carboxyl, then a carbonyl, okay, then an amino, or a phosphate, or a methyl. So 2, 3, 4, 5, 6, and then a sulfhydryl group or a thiol group. So these are your different functional groups. So any of these structures, any of these components can have any of these different functional groups in them. In this case, that we are going to talk about how a polymer is being made, or how monomers attach to give you a polymer. We are only talking about the hydroxyl group right now. Okay, so the hydrogen of the short polymer and the hydroxyl group of the new monomer that has come into picture together in the presence of an enzyme. Okay, please remember, all of these things, they just don't happen by magic. They definitely need an enzyme to act on it inside a cell, the right conditions, the right amounts of energy, and so on and so forth. So it removes a water molecule. Removal of a water molecule means dehydrating, and that will make it form a bond. So in short, let me have one guy here. Now this is a monomer plus another guy here, and these two guys coming into picture, losing a water molecule. Okay, that's what he's trying to show. So that process leads to formation of a long chain polymer. Now on the flip side, you can also lose the monomers or break it down to individual units. Okay, how is that done? That is done by a process called hydrolysis, okay, or breakdown. So this particular bond is being broken, and when this bond is being broken, you have to substitute it with water. So water gets split into H and OH, and on one side you will have H attaching itself and the other one OH attaching itself. So you split water, you added to these ends by breaking that particular bond, and they formed two new bonds of one with the hydrogen and kind of shortened itself by one monomer, and the other one had received its hydroxyl group back. Okay, so this is basically, in a nutshell, what is a polymer and how it is formed, or how you can get monomers from these polymers. So we have dehydration reaction and hydrolysis reactions. Okay, now we are going to talk about carbohydrates in detail. Okay, so my point is, before I talk about these carbohydrates, just wanted to give kind of like a heads up about my class, and once again my class, and putting it in perspective. What is the story behind it? So my class just started right after lunchtime, in which case everybody got themselves something to eat. So I start the lecture by giving everybody kind of perspective. What is the perspective I try and give is, you have eaten, so some take a Coke for themselves, and it has close to if my guess or memory is right, it has something like an 80 grams of sugar in it. That's what I heard a student say. I might be completely wrong. It had something like 80 grams of sugar in it. So that means that is sugar. Okay. So now when you have this sugar, it is a smaller molecule. Okay. And when it is taken in by your body, it finally enters the intestine, and then it is absorbed. Why? Because it can enter the cell, and it can enter the cell via blood vessels. Okay, so now this will be taken to different parts which need that sugar. Okay, so when they need that sugar, and just a second. Okay, now if they need that sugar, it enters directly. Okay, so now what is the next part? That now your body will take in as much as it wants, and then the rest of those sugars, it cannot just keep circulating in your blood, it has to be put away. So this is the time what happens is, what the rest of the sugar molecules, sugar molecules are undergoing a dehydration reaction, made into a polymer, and the polymer for us in our case is glycogen. Okay, and it is made into a polymer, the glycogen, and stored in the liver. There, done. Now, second option. I mentioned this because this was raised by a student, and so I thought it was relevant, and I'll put that also. Now, second option, you have bread. Now that is made of starch. Okay, now this starch is a large molecule, and so it cannot enter the cell. If it does not enter the cell, and now then what? It has to be broken down. So hydrolysis reaction is favored, wherein you split water and you take out one unit at a time, and that enters the cell. That enters the cell. If this is broken, then it enters the cell. So you have the hydrolysis reaction and the dehydration reaction in perspective, or how and where it is being used. Okay. So coming back to our story about the different types of carbohydrates. Okay. Okay. So sugars, you have monosaccharides. They can be of 3 carbon, 4 carbon, 5 carbon, 6 carbon sugars. Okay. And monosaccharide, the examples are glucose. This is a 6 carbon. Fructose and galactose. All of these three that are mentioned here are 6 carbon sugars. Disaccharide is sucrose, which is a combination of glucose and fructose. Then lactose, which is a combination of glucose and galactose. Okay. So how are these sugars structured? Okay, let's go to the next slide and see. So ideally speaking, if this is a sugar and C6H12O6, okay, so you have 6 carbons, 1, 2, 3, 4, 5, 6, and there are some hydroxyl groups to the right of the linear form and to the left of the linear form. This is your carbonyl group for you, and it is started, the numbering starts from the carbonyl group. Now, it is not favorable stoichiometrically for any molecule to be just one long linear chain. It is likely to come across a breakage and lose its structure. Okay, so it is more stable if it is able to turn, bond with each other in such a way that it can form a ring form, which is what is theorized here, wherein you can say the numbering starts from the carbonyl. So from the right you have 1, 2, 3, 4, 5 and 6. Okay, so this is your ring form. And also to be noted, all of the hydroxyl groups on the right side are falling down like a drop down stick. And this also is considered a drop down stick. Okay, all of the hydroxyl groups which are on the left kind of are on the up, kind of here above the ring. Okay, okay, so numbering is clear and the positioning of the hydroxyl groups is clear. It is important to understand this. The reason being, then you understand the two different polysaccharides that we will be discussing. One is starch and the other one is cellulose. Sorry. Okay, so starch is a group of glucose molecules. Bond. And cellulose too is a group of glucose molecules. But they are as different as can be because of the kind of bond that they make. Okay, so I normally make my students come out and stand. So then I have this one student stand here, and then another student stand here, and another student stand here. So I say this is the bond that's being formed. So it is the 1 of this particular glucose, and then if there is another glucose here, and the 4 of this particular glucose. You know what I'm saying? Okay, this one. So between the two they will lose a water molecule, and you will have kind of like a structure with this glucose here and this glucose here. Got it? So this is the 1 of this glucose and the 4 carbon of this glucose. So you have something called as an alpha 1,4. You can also have something like an alpha 1,2 or a beta 1,4, depending on the position and the way the glucose molecule is oriented. So in polysaccharides, the storage function, okay, that is, so polysaccharides have 2 functions chiefly, storage and structural, and the storage function is satisfied by starch in plants and by glycogen in animals. Okay, the starch have an alpha 1,4 linkage and they are invariably like straight, and so it's called amylose. If it is branched, it is amylopectin. In glycogen, it is almost always branched. On the other hand, cellulose, if you notice, it has a beta 1,4 linkage and they are kind of parallel. They are never branched, and so when they are held together by hydrogen bonds, that is holding the entire strand together, they also have the ability to absorb water. So your Turkish towels, okay, absorb a lot because they are made up of cellulose. Okay, so what do we know about cellulose? It is again repeating units of glucose bond in a beta 1,4 linkage. Their linkages or the microfibrils are parallel, they are never branched, and they form the dietary fiber. Where would you see it? And again, I bring to my class a leaf which is kind of dried up, and you can only see the structural framework of that leaf. Okay, kind of like a net you could see. Now this net that is being formed, that particular component that makes it form the net in such a way, is the cellulose, and that is responsible for the framework of the leaf. So you see, my storage polysaccharide is starch granules in plant polysaccharide, and in glycogen it is an animal polysaccharide. You would see huge big globs of glycogen granules present. Okay, so here it sort of gives you a perspective without putting any numbers. If you notice, your starch is, the glucose is oriented in the same way, and you have an alpha 1,4 linkage. Okay, so this is the 1 and this is the 4. So in cellulose, if you notice, the glucose is kind of upside down, so it is a beta 1,4 linkage. Okay, why is this important? Why am I taking so much pains to explain this? Remember, if there is a compound which is naturally occurring, there is an enzyme to break it down. And so things are brought down to their individual recycled simpler forms, like just carbon dioxide being released into the air and oxygen and all the other material being recycled back into the soil and so on. So my point is, if there is a compound, there is an enzyme for it. But if you notice, every one of us which can eat, consume, have an enzyme which can break down this alpha 1,4 linkage, and it's a happy story. But not so for cellulose. My meaning, if you have a salad which has a lot of leaves in it, mind you, that particular leaf, there is a big huge chunk of it that you are not really eating, but it passes through your system. Okay, so what is the use of this? Why do we need to eat it? Okay, so this is like one hard kind of stick, I would say gritty kind of material. It is able to scratch the insides of your intestine force it to secrete mucus, and so it can ease the passage of the waste. Okay, and that is fiber for you. Okay, now there is very few creatures in this world who have an enzyme to break down this beta-1,4 linkage, and those are some bacteria. But these bacteria are kind of sort of cultivated by certain creatures. Okay, so my point being, this is just structure of amylose, which is a straight chain, and then amylopectin, which is branched, and glycogen, which is ultra branched. Okay, so you have the different structures by difference in positioning of the hydroxyl groups, and so you will have a starch which is the 1,4, and this is alpha 1,4 linkage, and here if you notice it's beta 1,4 linkage, and the enzymes to break this down are rare. Not every creature possess it, only bacteria possess it. Some protists possess it. Some archaea possess it. Now, cows and other herbivorous animals possess a huge big chamber in their digestive system where they have this, they cultivate that. So they are the ones that are like one huge big fermentation tank, and they are able to break down cellulose and release the energy for themselves and also benefit the cow or the herbivorous creatures. Okay, so this is your cellulose, which is got these long fibrils, and because of that it is capable of forming hydrogen bonds and hold each other. And when you tear paper, you can kind of feel that it is fibrous and it is kind of in layers. Okay, that is one part of it. And in this slide, if you notice, you have the bacteria inside the stomach of the cow, wherein all of the food that it is eaten gets inside and it is slowly broken down. The cellulose part of it is slowly broken down by these microbes. Okay, so cellulose digestion is critical for herbivorous creatures. Okay, it's forming a source of nourishment. Okay, and the importance of fibers in human beings who don't have an enzyme to break down cellulose, but yet they have to consume a lot of fibrous material through plant material or leaves, lettuce leaves, spinach, and many other. That helps in easy passage of food and wastes. Okay, so inside the cow you would have a cellulose digesting prokaryotes and protists. They also are present in termites, and that is why they are able to eat wood. Okay, finally we have another structural polysaccharide which is seen in animals called chitin, and it is leathery and flexible. It gets hard when it is combined with calcium carbonate. You also see it in fungi. What is the economic importance of this chitin? You can extract that and convert it into the form of a surgical thread. That is what surgeons use if they want to do a quick stitch and let the stitches dissolve by themselves. Okay, so here if you notice, this is a molting insect. Okay, and this is the exoskeleton which has got chitin. You can extract that and make it into a strong flexible surgical thread. So in a nutshell, if I have I think I'm missing one page. Maybe it will come a little later. That time I will sum it up. Excuse me. Can I go back to the previous page for a minute? Okay. in a nutshell, okay, carbohydrates, carbs, are of two types, two functions that they have. One is structural and storage. Okay. In structural, you would have in plants cellulose, in animals you have chitin. Storage, in plants you have starch, and in animals you have glycogen. Okay. Okay. We move to the next macromolecule that we have to discuss in this chapter, lipids. Now, one very important thing about lipids is they are not true polymers, meaning they do not have no repeating units. Okay, they are just simply composed of fatty acids and glycerol. Okay, so in different combinations, right? So first thing is they are hydrophobic. I will go ahead and prove to you why they are considered hydrophobic in a while. Okay, so you would see lipids in pigments, in waxes, in fat, in phospholipids, and in steroids. So they are just large molecules assembled from other smaller, slightly smaller molecules. Okay. So this was the slide that I was expecting. I kind of misplaced it. So in a nutshell, for carbohydrates, the macromolecule that it is, okay, it is carbohydrate. The polymer of that is called a polysaccharide. The monomer of that is called a monosaccharide. The bond that connects each other is a glycosidic link, the alpha 1,4, the beta 1,4, and the beta alpha 1,2, and so on and so forth. Example of the carbohydrate, only one I'm giving here, is starch. And what is it composed of? Monomers called glucose. And the function chiefly is for storage, for energy. All right. So we move to the next part, the lipids. So, like I said, glycerol is an alcohol. So it's basically CH2, CH2, and 3 alcohol groups, that is the hydroxyl groups at its edges. Okay, so that's what this is. 3 alcohol groups it has, right? Then I want you to remember what our hydrocarbon was in the previous class. A hydrocarbon was carbon and hydrogen. Carbon and hydrogen. So if I am talking about a hydrocarbon, let me write. So if I can have CH3, CH2, CH2, CH2. They are bond, okay? CH2. And it goes on and on and on. At the end of it, it has one carboxyl group. Carboxyl group. Okay? So this carboxyl group that you have, that reacts with the alcohol group of glycerol. Okay? So the whole thing is kind of put the other way around. So if this is on this side and this is on this side, so it's sort of flipped, like you would flip a pen or flip a scale. Okay. So in this, if you notice, the alcohol and the carboxyl group, which means the OH and the COOH which is present between the two, they lose a water molecule, kind of like this, and that is called the dehydration reaction. In this particular case, what it ends up giving you is kind of like this. So this is an ester linkage. In some cases, in some books, it's also written as an acyl linkage. You can choose to remember either of these words. Okay, so your glycerol combines all of its hydroxyl groups with 1,1 fatty acid each, and they are all long chain fatty acids. Okay, now look at this particular molecule that we have and tell me if it has any part that has got a charge which can react, which is still unsatisfied kind of. Okay, nothing, because each of this bit, you have the carbon which is got its valency satisfied on all of the sides, so it is totally non-polar, no charge left, which means it is hydrophobic. Okay, if it is hydrophobic means it will repel water. So kind of, I would like to remind my class again of the lab that we just did, wherein we were talking about using one test tube with water, distilled water, and then layering it with vegetable oil, okay, or fat, and then if you mix it, they will separate out, meaning there is no way they will stay in suspension, so they'll go out into their individual layers. Excuse me. Okay, so you can have fats which are saturated and unsaturated. Saturated means it is completely filled with hydrogens and no double bond is necessary, which kind of packs it solid. There is no freedom for movement. Okay, so it is a solid at room temperature. The best example is butter or animal lard. Unsaturated fatty acids, on the other hand, have a double bond. My point being, if I'm able to go to the previous slide for a second, I'll show you. Here, if you notice in this particular slide, each of these carbon molecules that are present have hydrogens, enough hydrogens that they all make a single bond. Okay, and if at all I had less hydrogen, what would I do? At some point, if I had less hydrogen, I would be forced to make a double bond with a carbon. So then it will not have this kind of uniform structure that you see. Can you see that? The way it is organized, it's like very, very uniformed, kind of arranged in sheets. Okay, so that is not going to happen. So what is the outcome of having something like that, like a double bond? If I have a double bond, what would happen is I would have this guy, the rest of them would still be like this. Now tell me, do you see a proper solid structured packing in this? No, which means it will become or stay a liquid at room temperature. Okay, this is what I mean by unsaturated fatty acid. Unsaturated. meaning it does not have enough hydrogens to satisfy all the carbons to just be okay with just single bond. There is a need for a double bond if it is a unsaturated fatty acid. Okay, so that is with saturated and unsaturated fatty acids. So my point was, because it is unsaturated, it has a double bond, it kind of resists packing. So it stays a liquid at room temperature. Normally, plants, whatever plant oil that we see, is generally a liquid or an oil. Fish oil will also be liquid. See, it makes more sense because when it gets cold, it should not solidify. That means there would be no circulation. That will make it difficult. Okay. Also very important is cis double bond is favored over trans double bond. The reason being, sometimes you would lack, or naturally occurring, you would have cis double bonds and not trans double bonds. Okay, that's the first thing. Second thing is, many a times you would have artificial hydrogenation being done. My point is, butter is used for most baking and cooking, okay, and that has no double bonds, but it is expensive. So there was one product that is being released into the market where they artificially hydrogenate certain oils, okay, and make them look like solid and say, oh, okay, this is what you could use. Now with this, what happened was, it would revert back into its double bond nature, the unsaturated nature, back into the system. At that particular point, it ends up being a trans double bond instead of being a cis double bond. And this is not broken down, so it starts to accumulate inside the arterial tubes, leading to cardiovascular diseases and blockages called atherosclerosis. So this is what I mean when I'm talking about butter, where you could see it is saturated fatty acids that is present, and so there is very good easy packing, and so it is solid at room temperature. Whereas olive oil or any other oil would have an unsaturated fatty acid, and so it kind of has a kink, and so it will resist packing, okay, and that would cause it to be a liquid at room temperature. Okay. So what are the functions of fats? First obvious function is energy storage. Some of them are not made in the human body, so you sort of have to eat it from other sources, omega-3 fatty acids. Okay, and remember, 1 gram of fat has twice as much as energy that it can offer as much as 1 gram of polysaccharide. All right. Now, most plants are immobile, so starch can be the storage, and oil in the seeds is possible, that is, that they can deal with it. In animals, the energy storage is carried out in the adipose cells, and they need that as an insulation against the cold, so they need it to be a solid. And so the blubber that you would see in walruses or whale, okay, or seal, that is made up of saturated fatty acids, and so it will be a solid at room temperature, and harder solidified in case of colder conditions. And some amount of fat is extremely important because it sort of cushions the organs, all the different organs that we have. That is one. Another one which you have to be mindful of in any nerve cell. This is your axon, this is their cell body, and this is the dendrites. It is kind of covered by a material called the myelin sheath. This myelin is also made up of fat, okay, some amount of fats present in it. Okay, the second kind of lipid that we'll be talking about is phospholipid. Okay, phospholipid okay, so my point being, you would have phospholipids, which is comprised of 2 fatty acids, 1 glycerol molecule, and 1 phosphate group, meaning think E. That's what I tell my students. So our fat was a glycerol molecule with 3 fatty acids, right? Kind of like this. Now phospholipid on the other hand is glycerol molecule with 2 fatty acids and 1 phosphate group here. In this case, none of the sides or any part of it had any charge, so it was totally hydrophobic. Okay, but in this case, this part is hydrophobic, this part has a capacity to react because it is a phosphate group. Phosphate group have some amount of charges, so it makes it a hydrophilic region that you would have. Excuse the spelling. Hydrophilic region. Okay, so that was with respect to phospholipids. And where do you see phospholipid? It is found as a bilayer. Okay, one second. It is found as a bilayer. Okay, the phospholipid bilayer basically consists of a phosphate group, 2 fatty acids attached with a glycerol molecule. Okay, so the most common representation is kind of like this. So you have a hydrophilic head and 2 hydrophobic tails. Okay, and it is part of the cell membrane. And it is called a bilayer because you have 2 layers of these phospholipids arranged in a form of a sandwich, wherein the head is exposed to the outside and can react with material on the outside, and the head is exposed to the inside and can react with the aqueous part of the cell and react with the different materials on the inside. Okay. Going back to the previous slide, so steroids are the third kinds of lipids that we will be covering. One important part is the cholesterol. Now cholesterol is also part of the cell membrane in certain cells, and it is responsible for giving that cell either a certain amount of fluidity or certain amount of rigidity, and it kind of helps depending on which part of the world that particular plant cell contains it. So it can be having a broader range of tolerance over the cold. Okay. Steroids are also part of the hormones, or hormones can be classified, or some of the hormones can be classified as containing steroids. The example that we had already come across was testosterone or estradiol. Okay. Now, too much of cholesterol can cause a problem wherein it is not broken down and it stays blocking the smaller capillaries of arteries, and it can have a negative impact, giving rise to cardiac problems. Okay. Okay. So this is the structure of a typical steroid ring. The 3 hexagonal rings that you see is typical of a steroid, with one penta ring joined together is typical of the structure of a cholesterol. I'm sorry, I will reword it. It's typical of the structure of a steroid, cholesterol being one of them. Okay, so here I kind of end the section on lipids by summing it up. Lipids, I am talking about fats in this case. You can have mono parts of it, but you would not call it monosaccharide because neither the fat nor the fatty acid and glycerol are repeating units. They are just huge molecules they are put. So it is not a polymer. Very important to remember that. But they are made of these sub-smaller particles called fatty acids and glycerol. So you will have 3 fatty acids and 1 glycerol molecule with respect to fat. If it's a phospholipid, it's 2 fatty acids and 1 glycerol and 1 phosphate group, and they are joined together by an acyl or an ester linkage. An example can be butter. The glycerol and the fatty acids can be these different kinds: oleic, palmitic, stearic. These are long chain fatty acids. One of the function that we could remember is storage, and the other could be as part of the cell membrane or as part of hormone, and so on and so forth. Okay. Now we talk about proteins. Okay. Proteins is definitely a macromolecule and a polymer. This is definitely a polymer. Okay. Now he says 50% of the dry mass is protein. What does he mean by that? Supposing you have one person, okay, and you dry this person, remove the water. Now it sounds a little gory, but I'm talking about it so that you understand in perspective what this 50% means. So you completely dry, remove the water, and then you weigh this person. Whatever is the weight that this you get, half of that is contributed by proteins. That's what he means by 50% of dry mass. Okay. What are the different functions of proteins? Oh, proteins have a varied set of functions. One, they can act as enzyme and speed up a chemical reaction. They can be part of the structure and give structural support. Keratin, for example, is part of nail or hair. Okay, they can be part of storage. An example is ovalbumin. The egg white that you would see is the material on which the chick embryo depends on and derives its food in order for it to develop into a chick. Okay, then via the cell membrane there are certain proteins or certain material which are proteins, okay, and their function is chiefly transport. What do I mean by that? So if I have like a cell membrane, I might have a channel protein. okay, or a transport protein. This can help move a certain particle from inside to the outside. All right. So that is another function. The fifth function is it forms a hormonal receptor again on any cell. Supposing on the outside you have like a receiving station, and this is like capable of receiving the presence of a small hormone. So this particular material is made up of protein, so it is responsible for cellular communication. Then he says it is also responsible for movement, so contractile motor cilia. Okay, so what I am talking about is you would have this bacteria, okay, and it has a flagella, and this flagella will make this bacteria move. So this is composed of a set of proteins that act in a certain way to make this bacteria move. Not only in bacteria, in animal cells, the sperm cell, for example, has a flagella. It is composed of material, the material being protein. Okay. Also, lastly, it is the main item or main component of immune system, the antibodies. Now, I often have my students asking me, what is an antibody? At this particular stage, let me start by talking about when you have an infection. So you have, say, chickenpox. Okay. Now you have chickenpox and then you fall ill, you recover, you'll never ever get chickenpox again. Why do you say that? Because your body has made certain antibodies which are put in the memory, and next time when you get affected by this virus again, you have these antibodies which are ready to come and defend your body and deactivate this virus. So my point being, you have a bacteria. Okay, this bacteria has a certain material which is on the outside that can provoke an immune response. Okay, so it gets into your system and you then fall ill or don't fall ill and you recover from it. But then in your blood you have certain proteins called immunoglobulins. These are antibodies. Okay, these are made up of proteins. That's what he's talking about. Okay, now the polymeric form of protein is a polypeptide. So you have a polypeptide, which means it has individual monomers of amino acid. So I have amino acid 1 joined to amino acid 2 joined to amino acid 3 joined to amino acid 4. Now remember, in the case of carbohydrates, I was just talking about one glucose joined to another glucose joined to another glucose joined to another glucose and so on and so forth. In this particular case, it was the same molecule repeating units. In this case, it is repeating units of a certain kind or genre of molecules. Okay, so the monomers in standard study right now are amino acids. What is special about them? They are 4 different kinds of monomers that you will see. 4 different kinds. They are bound together by a linkage called the polypeptide linkage. Okay, and they have an amino terminal and an carboxy terminal. In order to understand that, just a second. Okay, so let's just take into consideration. I would like to go to the previous slide. Okay, in order to understand this, okay, I have a carbon, it has a hydrogen, it has an R group. I'm not even telling you what this is. It is a functional group. We'll talk about what this group is later. Okay, I'm just drawing a generic amino acid. On one side, it has an amino group. On the other side, it has a carboxyl group. Mind you, normally when I am writing a carboxyl group, I would write it like this, with the carbon attaching to whatever other material, like another carbon. So I cannot possibly show this like this. It is not the hydrogen which is attaching to it, it is the carbon that is attaching to it. And that is why I am putting it exactly en face or opposite, kind of like this, to represent it is the carbon part of the carboxyl group that is attaching to this carbon. So, point being, you could see that it has this particular carbon is called an alpha carbon or a central carbon. It has a hydrogen, it has an R group, it has a carboxyl group, it has an amino group. This is an amino acid. What is special about an amino acid? It has an carboxyl acid group and it has an amino group. Okay? Okay, so now we talk about how they are connected. How do they join in order to give me a long chain polymer? Okay, so let's draw one amino acid here. So I have, one second, let me just go back. I'm sorry. So I have, this is my amino acid. I am calling this the NH2 here, and I am calling this the COOH here, and I am calling this R1 because this is my amino acid 1 and this is my H, plus this is another of the, so I'm going to just put the H separately in order for us to understand this. So if you notice, I have drawn two amino acids where the only thing that is different between the two is the R groups. Okay, so if this amino acid joins with this amino acid in a certain bond, okay, what is that bond? They will lose a water molecule between the two. So it loses a water molecule. So there is a dehydration taking place. What do you get? Okay, what you get is, putting all of this as the same, I get a CO and an NH. This is the CO, this is the NH that got bonded here. This is the peptide bond. Okay. So that is what is represented here. Again, if you notice, he is talking about, now here what he has done is, he has already taken the liberty of showing you an art group, and I didn't want to do that to my class right now. I'm just asking you to think of this as one R group and this is another R group. This is my central carbon with an amino group and a carboxyl group. Amino group and a carboxyl group. Between the two, they lost a water molecule, and I ended up having a CONH bond. Right? This is a peptide bond or a peptide linkage. This is the bond that bonds the different amino acids together. So I would have a huge, big, long chain of amino acid 1 joined to amino acid 2, to 3, to 4, up to even 100, 200, 300 amino acids. Okay. Now, we are talking about the different R groups. Remember, in your standard amino acid, you have the carbon, you have the hydrogen here, you have the carboxyl group here, you have the amino group here, and you have the R group here, right? That R group can be just an H, and it will be glycine. It can be a methyl group, it can be alanine, it can be one of the trimethyl kind of group that has been attached to it, and it would be valine. And you would have many different kinds that can be attached. Okay, so if you notice, there is one sulfur group in this methionine in this. Okay, we'll talk about that later. Okay, I'll just remind you about that later. But if you notice, all of these guys, we have one very important thing to remember. Once they are bonded, that side will have no charge, will not react. Okay, and if this side and this side is already reacting to form the peptide bond, that is also gone. So an entire section, if I keep having these kind of amino acids, glycine, alanine, valine, all of them will have no charge and they will be hydrophobic. Okay, and insoluble in water. Okay, and so they are non-polar R groups. So they are amino acids which are non-polar. Okay, then you can have 5 to 6 amino acids which are polar. If you notice, the CH2OH has this OH which is capable of reacting. The CH3CHOH has this OH that is capable of reacting. CH3H can react, which it has got a sulfhydryl group. Remember, during the talk about functional groups, we were talking about sulfhydryl group and specifically about the amino acid cysteine. Now this amino acid cysteine, the interesting part is in any hair you would have a lot of cysteine. Supposing it is this amino acid which has this SH and an amino acid here that has this SH, it can okay, kind of sort of curl up and form a disulfide linkage which will make the hair curl, and this adds to a lot of curls. So it is said chemically, African American hair or any person who has extremely curly hair has got a lot of cysteine in their hair, the amino acid cysteine in their hair. Okay, now lastly we have if you remember, we have one carboxyl group and one amino group. If I have one more carboxyl group added, that means that amino acid is more acidic than basic. We have two of them like that, called aspartic acid and glutamic acid. On the flip side, if you have amino group here and a carboxyl group and another amino group, can you see? So that makes it more basic than acidic. So that's why you would have acidic and basic amino acids. So all in all, we have 20 amino acids, of which some are non-polar, some are polar, some are acidic, some are basic. So the non-polar ones generally are the part of any protein that doesn't really solubilize because it doesn't react with water. The polar part, the acidic part and the basic part are the ones that can react and make the protein soluble. Alright? Okay, now we go to the most important part of a protein, because till now we have been discussing only a polypeptide, meaning different amino acid 1 joined to amino acid 2 joined to amino acid 3 and so on, right? This is just a polypeptide. You cannot call it a protein yet. Why? The folding is what makes it special. How it folds, what parts of it is exposed, what parts of it is not exposed, what is the final structure of a certain side, that decides whether something else will come and bond with it. Okay, so you have to assume an analogy that I'm trying to give you is a polypeptide is like a yarn that is unfolded. But a sweater is something like a protein that is completely folded into its unique shape, final unique shape. Okay, so you could have fibrous insoluble proteins or globular soluble proteins. The fibrous insoluble proteins will have a larger percentage of non-polar amino acids present in it, whereas the globular soluble ones will have polar, acidic, or the basic kinds in them. So you have different structures and they are listed out in the gradual increase in complexity as the primary structure. The primary structure has got only a peptide bond. Okay, so what is holding them is the peptide linkage. So one amino acid joined to the other amino acid to the other amino acid and so on is that is your primary structure. The secondary structure, you have this particular guy kind of folded in an alpha helix. You can have two options in this. One is an alpha helix. What is the outcome, or how can it do this? There might be an amino acid here which has ability to form hydrogen bonds with the amino acid here. So you see, that is what holds them together. So I might have it like this and like this. So I'm just showing you the dotted part. So supposing this is the amino acid here and this is another amino acid here. So this might be amino acid 20. This might be amino acid 34. They can form a temporary hydrogen bond or interaction, and you can have an interaction kind of like that. Okay, that is your alpha helix for you. Or you can have beta pleats. That is also a secondary structure. Beta pleats meaning I have an entire polypeptide arranged like this, another polypeptide arranged like this, and they have certain charges that get them to make hydrogen bonds kind of like this, and that is going to make them be in sheets. Sometimes you must have noticed that certain meaty products, or you might sort of have them peel off in layers, and that's because of the beta pleats. Okay, this does not complete the story of the enzymes, protein structure. Why? They can further fold among themselves, which will be able to give you a tertiary structure. How does that work? Okay, so this is your alpha helix. If you see these dotted lines that you see, one second please, if you see these dotted lines that is there, those are the hydrogen bonds that can exist between an amino acid that is far away and another amino acid that just got closer and kind of held each other together. Okay, on the other hand, you can have them in sheets, and each sheet getting attracted to the amino acid in the sheet above. Okay, so you can have them that way. So beta pleats, an example is spider web, and the spider web strands are spiral, they are elastic, and they have these pleated sheets. This technically makes them stronger than steel with the same thickness. Okay, now talked to you about the primary structure and then the secondary structure. The tertiary structure is what we are dealing with. What am I talking about? Think about it. There is one amino acid which has this R group. Now can you see that this is polar, non-polar, I apologize, non-polar, no charge. This fellow is also non-polar. So ideally they should not even get attracted to each other, right? But what is their common enemy? Water is their common enemy. Because water is their common enemy, they avoid water and kind of come together and are held together by hydrophobic interaction. Then you can have van der Waals interaction. Then you can have further hydrogen bonds. You can have disulfide bridges like I told you, or you can have an ionic bond. So these are like, this might be amino acid 32, this might be 103, this might be amino acid 246. I'm just giving you example randomly. This might be amino acid 320. This might be amino acid 465. So can you see that this kind of bonding makes the protein fold up in different ways and kind of come together as one huge big glob of a final shape and not just like a strand. And this final shape is all naturally determined. You and me cannot decide this. Okay? Now, tertiary structure was the further folding after primary and the secondary structure, which is what I explained in the slide before. Quaternary structure basically means this is one polypeptide which folded up. This is another polypeptide. This is polypeptide A. This is polypeptide B. There's another polypeptide C. All of these guys together now have come together and have formed a huge big glob, which is quaternary. So sometimes you would just have a tertiary structure and that is your final protein. Sometimes some proteins have more than one polypeptide, and that makes them quaternary. Example, you can see collagen has 3 different polypeptide chains in a braid, okay, and hemoglobin has 2 beta chains and 2 alpha chains, and they have like an iron or a heme protein, the heme that sort of holds them together. So these are this is one unit, this is another unit, this is another unit, that's another unit. All of these guys have come together to give me one entire hemoglobin. Okay. Just a note, if there is a mess, this particular topic will be like kind of repeating 2-3 other times in 2-3 other chapters. Okay. Just a note to say is hemoglobin has a primary structure. So if you notice in the 6th amino acid, it has glutamic acid. All right. Now, instead of glutamic acid, if there is a mutation and the cell puts valine instead of glutamic acid, it puts valine. It messes with the structure completely and you end up getting a red blood cell which has messed up hemoglobin, and that red blood cell is called a sickle cell. So you can see that just one amino acid can mess with the entire protein folding and mess with the outcome of that particular protein, which ends up being defective. All right. So protein folding is an extremely serious business, and it's very, very important that proteins are folded in the right way in order for them to be functional. All right. Okay. Now, you also have another property to discuss. Proteins can be denatured and renatured. Some, okay? Meaning, think egg, okay? Raw. So you have the okay, let me come back. Okay, so you have the normal protein, so think egg, raw, wherein you have the yellow and the colorless part of the egg. You would break that, put it into a bowl or put it into a hot pan, okay, and so you would get it all white, and this will also become a little different kind of yellow. Okay, this tells you the protein is not destroyed. Okay, the protein is there. What is destroyed? The structure is destroyed. So the protein, meaning the polypeptide, the primary structure still exists. So the peptide, the primary structure, it still exists. Nothing is messed up with the primary structure. It is the secondary, the tertiary, the quaternary structure that has gotten messed up in proteins, and that can completely change the outcome or the way in which the protein would behave. So different things can cause this particular situation. What are the different things that can cause this situation? Okay. Heat. Okay, so you can have heat that can cause a change in structure, or temperature in general can cause a change in structure. Salt content can cause, or if you have some amount of organic solvents, that can change the. Now, sometimes some structure can be renatured back. Some are permanent, like heat mostly causes permanent damage to the protein structure. Okay. Another part of this chapter is we can in the lab find out what a polypeptide is made up of and make a polypeptide. But it is very difficult to simulate the folding, the natural folding. We are still learning about the whole natural folding process. Okay, and the folding happens in, so far where we have reached is they have identified a particular organelle called chaperonin. It looks like a hollow cylinder with a cap, wherein the polypeptide which is being made by the cell enters, and it's like a changing room where a lot of magical things happen, where the protein gets folded and the cap falls off with the correctly folded protein being released. Okay, so this particular organelle is called a chaperonin. So to sum it up, proteins is the macromolecule, polypeptide is the polymer, that's what it's called, and the monomers are called amino acids. The bond that connects them is the peptide bond or the CONH bond. And the example, one example that we used in the lab today also was the albumin and different amino acids. You have 20 different amino acids that can be monomers, and one of the function is storage. But I already listed to you in the beginning of the slide for proteins easily about 7 to 8 different functions for different proteins. Okay, we move to the next topic of nucleic acids. Okay, so nucleic acids, remember, of all of the different macromolecules that we have studied, this is one new macromolecule that we do not have to constantly be supplied with. Our body can make it. Okay, so but there are dietary sources where we can get some amount of nucleic acid. One is mother's milk, some nuts, some fish, mushrooms, single cell protein. All of these are natural sources of nucleic acids. Best example of nucleic acid is DNA and RNA. Okay, DNA is the source of information. RNA translates and makes sense out of that information. Okay, the basic structure is it has certain components, that is a nitrogenous base, a phosphate group, and a 5-carbon pentose, that is sugar. Okay, a nitrogenous base, a phosphate group, and a 5 carbon sugar. These three things are the units that make one monomer or one nucleotide. Okay, so the nitrogenous bases that we were talking about, you have 5 of them. Some of them are double ringed. You can see that they have 2 rings in them, and some of them are single ringed. Okay, the double ring fellows are called purines. The single ringed fellows are called pyrimidines. You have 2 purines, adenine and guanine, and 3 pyrimidines, cytosine, thymine and uracil. Okay. The 5 carbon sugar that we are dealing with, in DNA you have deoxyribose sugar, in RNA it is a ribose sugar. Okay, both of them are 5 carbon, and the numbering starts from the extreme right, and it's called 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, so 1, 2, 3, 4, 5. Each of these notches represents a carbon. Okay. Okay, so before I proceed to this bit, this is just to explain how they are being organized. Okay, so in a polymer or a polynucleotide, what you see is you have a sugar, then a phosphate, then a sugar, then a phosphate, then a sugar, kind of like a backbone. So it starts with a phosphate, a sugar, a phosphate, a sugar, a phosphate, a sugar. So here he's made it all this squiggly lines. I would rather that you remembered it just as a straight line. It's fine by me. So you start with a phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, so this is how you would have a backbone. Okay, to this sugar on another part is attached the base. Okay, so just the base and the sugar can be called a nucleoside. Remember the S. Okay, for a nucleotide you have the base, the sugar and the phosphate molecule tied, so 3 items together. So each monomeric unit of a polynucleotide is called a nucleotide, and it has got 3 things. So the way it would be is, I have a phosphate, I have a sugar which is kind of attached like this, and here I have a base, a nitrogenous base. So you see a phosphate group, a sugar and a base, nitrogenous base. These 3 things comprise a nucleotide. Okay, so differences between DNA and RNA. Okay, DNA is called deoxy ribose or ribo, I apologize once again, ribonucleic acid or DNA, and RNA is called ribonucleic acid, RNA. Okay, The differences between the two. DNA, it has two strands of DNA that will make it up as a complete entire entity. RNA has only one strand. The sugar that is present in DNA is deoxyribose. In RNA, it is ribose. The bases, the nitrogenous bases that you would see is A, T, G, and C. Adenine, guanine, thymine, and cytosine. Okay. In the other hand, if you notice, for an RNA, you have adenine, no thymine. It has uracil, guanine, and cytosine. Okay, and you have only one kind DNA, double strand, that's it. You have three kinds of RNA depending on the kind of function they have: mRNA, rRNA, tRNA. Okay. Okay. mRNA is also called messenger RNA. rRNA is also called ribosomal RNA. tRNA is also called transfer RNA or translational RNA. Sometimes it is called translational. In some places it's called transfer RNA. So, ideally, adenine and thymine of the opposite strands of DNA. So, I have like an adenine here and a thymine here in the two strands of DNA. They can form a bond. It is like a temporary hydrogen bond. Okay, it's like an attraction. Their chemical structure is aligned in such a way that they can form two hydrogen bonds. Okay. Between guanine and cytosine, meaning this is guanine here, this is cytosine here, it can form three hydrogen bonds. Okay, so that is with respect to the bonding between the nitrogenous bases or the nucleotides of the opposite strand. Okay. Okay. Then another thing to remember is DNA structure is anti-parallel. What I mean by anti-parallel is, think about a street. You have this street and you have like a division. In this side, you have cars going in this direction, and in this side, you have cars going in this direction. So this goes this way, this goes this way. Yeah. So that is what it means by anti-parallel. So let's put it in perspective with respect to what your DNA should look like. Okay. So to understand that, the DNA strands, okay, remember, have this deoxyribose or this ribose sugar. Okay. So the ribose sugar, I would like to go back a few pages and explain that for a minute. No, one second. Okay, let's take the example of this ribose sugar. So, if you notice, if I'm taking this particular deoxyribose here, okay, so I have an oxygen here, it's a 5 carbon, okay, this is my first carbon and it has a hydroxyl group here, okay. This one does not. This is my second carbon. It doesn't have anything. The third carbon has a hydroxyl group which is a drop down. And the fourth one doesn't have any hydroxyl groups. The fifth one does. This hydroxyl group that is present in the fifth carbon. Right? Now, what happens is, if I have like a phosphate attached to a sugar and then to a base, how is the attachment taking place? One hydroxyl group, which is in the 5th carbon position, attaches to a phosphate group. Got it? It reacts, forms a bond and attaches there. The other hydroxyl group that is present in position 1 carbon attaches to the nitrogenous base here. So what do I have free? Only this drop down 3 prime hydroxyl group that is ready to attach to something else. So I can attach another phosphate group containing nucleotide in this fashion. So this bond that takes place with this nucleotide and the next nucleotide that came into picture is called the phosphodiester linkage. Okay, they lose a water molecule and I have a linkage there. But what is to be noted? This is in the 5 prime end and I have just this drop down 3 prime left. So the attachment of future nucleotides always happen in one direction. So if I have a strand which has got 5 prime, I will only have attachment or more attachment in the 3 prime end. I cannot have anything attaching here. That does not take place. That is the most important part. And then the way they are aligned is, okay, one second. The way they are aligned is, I would have which means this is my 5 prime, this is my 3 prime, this is my 5 prime, this is my 3 prime. So this is how you would have them aligned as an anti-parallel structure. Okay. And what is the meaning of complementary? Okay, complementary means if I have a strand of DNA which has got A, G, G, C, D, T, A, G, and the opposite strand DNA will have C, T, A, G, C, C, T. Do you see that it is not exactly not a copy, but it is the like a negative of, like a negative film that you would say. It is exactly like a mold. Like for example, you take a penny and you take some clay, polymer clay, and you make a mold. So the penny has made a mark on that clay. That mark is not identical to the penny, but you can get the penny if I am dropping some amount of like say like a polycarbon glue or something, and I put in that and I will get the shape of the penny again in that. Does it make sense? So I can make it like a mold, meaning one strand can serve to make the other strand. So it has the information to make the other strand. So this is like one of the body's foolproof mechanism, play safe mechanism. If I have some parts of it lost, gone, cut, something happened to it, mutated, I have just this backup part from which I can make this part again. Got it? Okay, so that is what he means by the anti-parallel nature and complementary nature. Okay, so to sum it up, nucleic acids, okay, the polymer is called a polynucleotide. Monomer is a nucleotide. It has 3 things, remember. It has the base, Please do not say it has adenine, guanine, thymine, cytosine and uracil. That is not the right answer. It has three things. It has the base, it has the 5-carbon sugar and a phosphate group. Okay, so if I am writing dATP, dCTP, dGTP, dTTP, I am basically saying it is deoxyadenine triphosphate or deoxycytosine triphosphate. It can be triphosphate or monophosphate, whichever one. Alright, so my point is this comprises of three things which make it up. If you just write the monomer is A, T, G, C, you are wrong. Okay, and the linkage that connects them, like I walked you through, is the phosphodiester linkage. Example can be DNA or RNA, and the function can be source of information or to translate that source of information. Okay, this kind of sort of sums up what the entire chapter was all about. So you have the carbohydrates where you have one monomer and then the different polymers listed out and what are the different functions. This is like the entire chapter in a nutshell. It's a good idea to go over each one of them. So this tells you that this is like a fat which is comprised of glycerol and fatty acids, the phospholipid and then the steroid hormone. Alright. And you have proteins. Remember, you have 20 different types of amino acids. The different examples that you would have with their functions is it serves as an enzyme to catalyze chemical reaction, as a structural protein to provide support, a storage protein to store the amino acids for like development of a chick, for example, or transport protein to move the substances across, or hormones to give some information or connect or pass a message. Receptor protein is to receive something from outside the cell. Motor proteins is for movement. Defensive proteins is like the antibodies that I was talking to you about to protect against disease. Nucleic acids store and transmit hereditary information. So here they have like a nucleotide monomer. Okay, if you notice, it has the phosphate group, a sugar and a base. It can be double or two ring base or the single. So this can be A, T, G or C. The whole thing is not the whole thing, or the monomer for that nucleic acid is not ATGC. Please be mindful of that. Okay, and their function is hereditary information or protein coding instructions are followed. Okay, this is a slide that you should know even in your sleep. Okay, it sort of sums up the different components, their polymer and the monomer and the kind of linkage. Okay, that's all for this. Thank you. Bye bye.